This report details the top 5 RNA chemical modifications, their IP positioning, market landscape evolution, economic potential, and short-form due diligence. It also includes competitor mapping by indication and an economic valuation forecast for both commercial and clinical stage assets. RNA therapeutics have evolved rapidly in the past decade. In this report, we discuss the top 5 RNA chemical modifications driving this progress, including the foundational technologies underpinning them and how they're being deployed across various modalities and indications. We analyzed thousands of data points to identify the most important RNA IP assets, which are detailed here along with our short-form DD, competitive landscape, and an economic valuation forecast for both commercial and clinical stage assets. Let's get into it. No. 1. On 1-methylpseudouridine MC. And 1-methylpseudouridine is a modified uridine analog that reduces immune activation and enhances mRNA translation. It's widely used in mRNA vaccines, including those by Moderna and Pfizer, BioNTech, which were foundational to COVID-19 vaccine development. Number 2. On 6-methyladenosine MBA. And 6-methyladenosine is an epitranscriptomic modification that regulates RNA metabolism, stability, and translation. It's targeted in cancer and regenerative medicine for its ability to modulate gene expression at the RNA level. Key players include Storm Therapeutics, who are developing a metal-3 inhibitor in early-stage oncology trials, Accent Therapeutics, who are focused on M6A modifying enzymes for ML and glioblastoma, and Epibiologics, who are in preclinical work for immuno-oncology. IP for M6A covers RNA methyltransferases, for example, metal-3 methylpseudouridine inhibitors, and use cases. Key academic IP has been licensed from institutions like Stanford and Chicago. First-in-human trials for metal-3 inhibitors are underway. Current asset valuations are between $400 to $700 million, with potential to exceed $2 to $3 billion if Phase 2/3 success is achieved by 2027. No. 3. 2-O-Methylation 2-OM. 2-O-methylation is a modification at the 2-hydroxyl group of the ribose that stabilizes RNA and prevents immune activation. It's the most widely used modification in ASOs and siRNA, often combined with phosphorothioate backbones or GalNAc conjugates. Key players include Alnylam Pharmaceuticals, who have 3 commercial products, Onpattro, Givlaari, and Oxlumo. Ionis Pharmaceuticals, who have SPINRAZA and over 20 pipeline assets, and Arrowhead Pharmaceuticals, who are focused on GalNAc-based RNAi therapeutics. IP for 2-on-sugar chemistry is extensive and mature, covering various sugar modifications, conjugates, and sequences. This results in lower FTO risks compared to newer chemistries. siRNA and ASO products with 2-O-Me generated $5 to $7 billion per year, with a projected 15 to 20% CAGR through 2026. Number 4. 2. Fluoro 2F modification. The 2-fluoro modification replaces the 2-hydroxyl with a fluorine atom, enhancing binding affinity and stabilizing RNA duplexes. It's used in siRNA and stereopure ASOs to fine-tune efficacy and duration. Key players include Alnylam Pharmaceuticals, who incorporate 2F as part of their core siRNA designs. Wave Life Sciences, who are targeting CNS diseases with stereopure oligos, and Silence Therapeutics, who are using extrahepatic siRNA with GalNAc conjugates. IP for 2-fluoro covers sugar chemistry, stereochemistry, and targeted design. Wave has proprietary control over stereopure backbone designs. Wave's valuation is currently tilted $700 million, with first CNS programs in PHI2 for Huntington's and ALS. Silence's cardiometabolic siRNA SLN360 is entering Phe. If approved, this could represent $2 to $4 billion in annual sales. Total valuation of these clinical stage portfolios is currently $1 to $3 billion, depending on success. Number 5. Self-Amplifying RNA saRNA. Self-amplifying RNA is based on alphavirus replicon systems. As RNA can replicate intracellularly, generating high protein levels at low doses. It's a promising next-gen RNA vaccine platform, particularly attractive for infectious diseases and oncology, where low-dose durable response profiles are desirable. Key players include Arcturus Therapeutics, with their COVID-19 saRNA vaccine ARCT-15 for approved in Japan and a pipeline in flu and RSV. Vaxequity, the Imperial College spin-out, partnered with AstraZeneca, CureVac, who are transitioning to the saRNA platform following their COVID failure and developing an RSV vaccine. IP for saRNA includes replicon design, immunogenicity modulation, and delivery systems. Strategic partnerships, e.g., Arcturus with CSL and Vaxequity with AstraZeneca enhanced development capacity. ARCT-15 is filed globally with decision expected in coming months. Asset valuation is currently $500 million and is expected to increase to $3 to $4 billion depending on regional approvals and pandemic preparedness strategies. We hope you enjoyed this report. Stay tuned for more updates and feel free to reach out with any questions.